everybody, and thank you for your attendance to this webinar. It's an honor and a pleasure for me to speak about this topic. In this talk, I will speak about binding and mycotoxin adsorption, and I will give you the options that we can have in these fields of science. Toxins are secondary metabolites of some microscopic fungi. They are not associated with growth or primary metabolism. Their biological meaning is not completely clear. They are produced in optimal conditions of temperature and humidity, and they are stable to the physical, chemical, and biological processes of food processing. The problem is that mycotoxins are toxic for humans and animals, and they are able to induce mycotoxicosis. In this slide, you can see the chemistry of the most important mycotoxins that you can find in foodstuff. They are not a unique chemical class. They have a relative low molecular weight, a moderate or high polarity, low volatility, and high stability. Each mycotoxin is produced by one or more fungal species, and in some cases, a fungal species can produce more than one mycotoxin. Here, an overview about the presence of mycotoxins in foodstuffs. As you can see, the most of mycotoxins are present in corn byproduct, in cereals. There is aflatoxin M1 that is a big problem in milk and derivatives. And the problem usually is that there will be a co-occurrence of mycotoxins in a lot of foodstuffs. There are different decontamination detoxification strategies that have to have in common some features. They have to inactivate, destroy, or remove the mycotoxin, avoid the production of toxic residues in the final products. An important point is that they have to retain the nutritive value and food feed acceptability of the products, preserve technological properties, and be economical, simple, and not too time-consuming. Among these strategies, you can find physical strategies, such as removal of fines, screenings, gamma radiation, and so on, the use of different chemical compounds to reduce mycotoxin contamination, biological strategies, such as microbial degradation, and feed food processing, so thermal and mechanical treatments applied to raw materials. The problem is that these strategies show different drawbacks. For example, physical treatment are usually expensive with uncertain results, often connected with high feed food losses, so with limited practical application. Concerning chemical treatment, they are expensive too, usually changing palatability and the nutritive value, and the important point that toxic byproducts are possible, so no practical application. An alternative to these strategies, there are feed additives. Feed additives, European regulation in 2009 established a new functional group of feed additives in the category of technological additives. These feed additives have to suppress or reduce absorption, promote the excretion, and modify the mode of action of mycotoxins. Anyway, no increase of the existing max guidance levels of mycotoxin in feed are done. Anyway, to put in the market feed additives, it needs an authorization procedure. So the applicant needs to prepare and submit a dossier according to the Efsa guidelines. Efsa is responsible for the evaluation of the data and can provide a positive opinion. After this positive opinion, this product, the feed additive, can be released in the market. In this slide, you can see the 3 feed additives regulated in Europe as mycotoxin detoxifying agents. First one is the bentonite, used as feed additive for ruminants, poultry, and pigs as aflatoxin binder. The second one is a microorganism to biotransform trichothecins for pigs and all avian species. And the third one is a group of regulations concerning an enzyme for degraded fumonisins in contaminated feed. Of course, other feed additive can be registered in this regulation if the applicant wants to invest some extra cost to register his product. Concerning the requirements of the assessment of the mycotoxin detoxifying agent, we need to characterize the additive by some instrumental methods. We need to assess the safety of the feed additive, in particular for mycotoxin binder. We need to assess that no adverse effect on digestion and absorption of nutrients happened. And of course, we need to evaluate the efficacy. So to specify for which mycotoxins additive will exert its function and target species, and to declare and demonstrate the mode of action of the additive, if it's an adsorption, for example, or a biotransformation. Efficacy assessment can be done by in vitro study, a very potential tool to screen different mycotoxin detoxifying agents and to study the mode of action of the additive. But this not mimic the conditions of the digestive tract. So Efsa in Europe required in vivo studies to demonstrate the efficacy under practical conditions by short-term studies in target species. The analysis of mycotoxins and the metabolites can be done using the biomarker approach, so evaluating mycotoxin excretions in feces, urine, or concentration in blood, plasma, and serum, or in tissues or animal products. Concerning the methods to assess the efficacy of the product, we can have in vitro study, and inside this study we found the adsorption trials using a single concentration of mycotoxins, adsorption trials using different concentration of mycotoxins or different adsorbent dosage of the product. So this is the famous methods of the isotherms. We can also study the desorption processes to understand the strength of binding between our product and mycotoxins, and to understand the more specific physicochemical properties of these processes, we can study also the thermodynamic of the adsorption. We can use for efficacy assessment also gastrointestinal models, static or dynamics, and in the end, in vivo study, studying non-specific parameters such as the state of the health of the animal, weight of the animal, and so on, or specific parameters, the biomarkers approach. What we can say about the vitro in vivo comparison? Both of these trials show advantages and drawbacks. Concerning in vitro advantages, welcome to everybody, and have organics binders such as yeast cell wall, agriculture byproducts, or other organics polymers. Inorganics binders, the most famous clays, and synthetics binder, in particular modified minerals such as organoclay, activated clay, or mixture between organic and inorganic binder. More than 100 commercial products are available on the market. Most of them claim the ability to adsorb several toxins, but simultaneous multitoxin adsorption has been poorly addressed. Okay, we can start with an overview about organic binders. I want to show 3 examples of this kind of binders, starting from yeast cell wall. These in vitro studies show how yeast cell wall used at 2% of weight per volume dosage adsorbed more than 85% of zearalenone, ochratoxin A, and fumonisin B1 at pH 3 and 7 used to simulate the gastrointestinal tract of the animal, whereas aflatoxin B1 absorption was lower than other mycotoxins. How we can explain the adsorption mechanism on this kind of binder? There are different papers in literature that explain this topic. I selected 2 interesting papers in which the authors take into account the complex structure of yeast cell wall that is characterized by polymers of mannose, monoproteins, beta-D-glucans, and other organic compounds. They found how beta-D-glucans are the yeast component responsible for the complexation of zearalenone. Weak hydrogen and van der Waals bonds are involved in this chemical complex formation. The authors extend these results also to other kind of mycotoxins. In the right part of these slides, there is a work of Yannikoulis et al., in which they found a correlation between the amount of beta-D-glucans in cell walls and complex forming efficacy. Increasing the amount of beta-D-glucans in yeast cell wall, you can find an increase in the amount of zearalenone adsorbed by yeast cell walls. This product was also tested in vivo using 24 sexually mature girls that were fed with 3 diets for 42 days. First diet was the control, second diet was the naturally contaminated diet with zearalenone, And the third diet was the naturally contaminated diet, supplemented by each cell wall product at 0.2% of dosage. Urine samples, 24-hour post-administration, were collected, and the amount of zearalenone and metabolites were analyzed using the biomarker approach. This work is in submission in a scientific journal. What about results? As you can see from this box plot, this graph, at day 42, the group supplemented, the group of piglets supplemented with yeast cell wall showed a 68% of zearalenone and its metabolite reduction comparing to the group without yeast cell wall product. This reduction was statistically significant, and this means that this product was able to reduce the pig's urine excretion of zearalenone and metabolites. Okay, moving on the second examples, I want to speak about agriculture byproduct, in particular about grape pomace. This product was obtained inside the big European project, MycoRed 7th Framework Program, and we found with some in vitro studies that using this product at 2% weight per volume of dosage, grape pomace absorbed more than 90% of aflatoxin B1, zearalenone, ochratoxin A, despite 2 pH values used, whereas fumonisin B1 absorption was lower than other mycotoxins. Same product was also in the same project, European project, was tested in vivo in order to assess the ability of this product to reduce mycotoxin bioavailability in piglets. As you can see from this graph, grape pomace reduced significantly urinary mycotoxin biomarker of aflatoxin B1 of 67% reduction and zearalenone 69%, whereas reductions statistically not significant were observed for fumonisin B1, deoxynivalenol, and ochratoxin A. So these are very interesting results. More details about this product, about in vitro and in vivo trials, can be found in these two recent papers published in the Journal of Agriculture and Food Chemistry. Concerning the third examples of organic binders, I want to speak about Durian Pill, and in particular about its modification. This work was done in collaboration between Institute of Sciences of Food Production and Thammasat University in Thailand. Why in Thailand? Because this is a typical fruit in Thailand. Its waste was more than 200 tons per year, and it caused environmental and social problems in Thailand. So we tested this product as such, and we performed also an acid hydrolysis on this product to obtain a modification of this product called acid-treated Durian Pill. After that, we evaluated the multi-mycotoxin adsorption activity. What about results? In these graphs, you can see that acid-treated Durian Pill, red bars, showed high adsorption values towards aflatoxin B1, zearalenone, ochratoxin A, and fumonisin B1. Non-adsorption was negligible, and the pH affected ochratoxin A and fumonisin B1 adsorption. Anyway, the modification of this product of natural Durian Pill increased the mycotoxins adsorption. This acid-treated Durian Pill was also tested with a gastrointestinal static model following the Mynekus et al. Protocol that can be found in the paper published in 2014. And we studied the capacity of this product to reduce the bioaccessibility in the gastric and intestinal phases of these 4 mycotoxins. As you can see from the results, acid-treated Durian Pill reduced the bioaccessibility of mycotoxins in gastric phase, and in particular, aflatoxin B1 and zearalenone showed a reduction in the bioaccessibility also in the intestinal phase. Also for this product, for more details, you can read this interesting paper published in 2020 concerning the effectiveness of Durian Pill as a multi-mycotoxin adsorbent. Of course, this is not an exhaustive list about organic binders and about agricultural byproducts. And other information about comparison of different kinds of agricultural products to be used for mycotoxins adsorption can be found in this other work published in 2019, Comparative Efficacy of Agricultural Byproducts in Sequestering Mycotoxins. Okay, moving on to the second category of binders, I want to speak about inorganic binders, in particular about clays. Clays belonging to the phyllosilicate class and to the smectite family, because these are the most inorganic binders studied in the literature concerning the mycotoxins adsorption. These clays are characterized by different sheets of tetraedric, octaedric, and tetraedric structure, TOT sheets, and the smectite family can be divided into deoctaedral smectite and trioctaedral smectite. Inside the deoctaedral smectite, we found montmorillonite, very famous minerals to be used for mycotoxin adsorption. And inside the trioctaedral smectite group, we can have, for example, saponite or hectorite. Among the smectites, deoctaedral or trioctaedral, we can find bentonites. Bentonites are clay minerals belonging to the phyllosilicate class and to the smectite group. I want to speak about bentonite in particular because in Europe, bentonite is authorized as a feed additive, in particular as a substance for reduction of the contamination by aflatoxins for ruminants, poultry, and pigs. Bentonite received the claim 1M558 to be used for this issue. But to be obtained this claim, regulations say that bentonite has to show more than 70% in smectite percentage, less than 10% in other minerals contaminants, and this smectite has to be in the form of deoctaedral montmorillonite. And above all, after a specific adsorption assay described in the regulation, aflatoxin B1 binding capacity has to be more than 90%. So we can explain the mechanism of adsorption between smectites and mycotoxins, in particular aflatoxin B1. There are two most accepted suggested mechanisms for this kind of process. One is electron donor acceptor, and the other one is potential chelation. Electron donor acceptor, in this mechanism, authors explain how smectites are negatively charged due to isomorphic substitution in this structure, and so they can attract positively charged ions to balance this charge. These positively charged can be the carbons in the decarbonyl system of aflatoxin B1 that are partially positive. Moreover, aflatoxin B1 is planar, so can go inside the interlayer space between one sheet and another one in the phyllosilicate and can be adsorbed. So interlamellar region, from this mechanism, interlamellar region is the primary site of the binding of aflatoxin B1. Instead, external surfaces accounting for only minor adsorptions. And external surfaces can explain also the adsorption of other kind of mycotoxins that present a structure different from aflatoxin B1. Potential chelation explain how mycotoxins, in particular aflatoxin, can be chelated by some cations present in the interlayer space of the smectites or by some various edge site metals. Anyway, not all deoctaedral smectites are the same. In a recent study performed in a big European project, MycoKey project, we found how smectites, aflatoxin B1 adsorption from smectites can be affected by geological origin of these minerals. We divided smectites in two groups, in sedimentary group and hydrothermal group, concerning taking into account their geological origin, and we performed adsorption isotherms on these products. As you can see from these box plots, we found that sedimentary smectites showed significantly higher values for maximum adsorption capacity and adsorption affinity than hydrothermal smectites. So, geological origin can be an important topic to see, to use this product as aflatoxin B1 adsorption. For more details concerning the role of geological origin and their physical-chemical properties on aflatoxin adsorption, you can be observed, you can be read this paper published in 2019 on the Applied Clay Science Journal. Natural smectites are able also to absorb fumonism B1. But as you can see from the graph, this adsorption is affected by pH. In particular, fumonism B1 can be absorbed at pH 3 and with a less extent at pH 5, but absorption at pH 7 is negligible. Here you can see an example of a montmorillonite clay used in vivo in rats. The authors, Mitchell et al., explain how this product, this commercial product, is able to reduce aflatoxin B1 and fumonism B1 biomarkers in rats exposed to single and co-exposures of toxins. They study pharmacokinetic excretion of aflatoxin M1 in urine and fumonism B1 in urine of rats, and they found a significant reduction in bioavailability of aflatoxin B1 and fumonism B1 in this animal model. So they conclude that this product may be used to reduce toxins levels below the thresholds for affecting adverse biological effects. Okay, in this slide, I want to focus the attention about the deoctahedral group and the trioctahedral group of smectite. Even if the regulation explained how smectite has to be in the form of deoctahedral structure, in our experience, we found how, following the European regulation, the absorption assay described in the regulation, also other kind of smectite, in particular trioctahedral smectite, can be fulfill the regulation and can be reached the 90% of aflatoxin B1 binding capacity. So, seems that this kind of group of minerals can be improperly excluded by the European regulation. Here you can see an example of the use of a trioctahedral smectite in vivo. This is a big Italian project, farming project, and in this project we studied the capacity of this trioctahedral smectite to reduce aflatoxin M1 amount in cows, in bulk milk, cows exposed to aflatoxin B1. As you can see from this graph, the group of cows treated with trioctahedral smectite showed a significant reduction in the aflatoxin M1 concentration and aflatoxin M1 total excretion in bulk milk. So this product seems to be a promising product, a promising minerals to be used in animals. Okay, moving on the last part of binders, the third categories, I want to speak about synthetic bentonite-based material. And I want to spend a few words on organoclay that are very famous synthetic materials. Organoclays are characterized are a mixture between natural smectite, natural bentonite, that are treated with an quaternary alkylammonium ions. These quaternary alkylammonium ions can go inside the interlayer spaces of smectites and can be characterized and can be obtained a new organophilic compound to be used for different issues, such as mycotoxin absorption. As you can see, these three examples of organoclay, you found a very good mycotoxin absorption values for aflatoxin B1, zearalenone, ochratoxin A, fumonism B1, and T2 toxins, despite two pH values used. Deoxynivalenol absorption was negligible also for this product. Concerning carbon, activated carbon, or mixture between bentonite and carbon, these are very good in vitro products. They are able to bind almost 100% of all mycotoxins, and for activated carbon, also DON is absorbed on these minerals. These two categories of product are very good as in vitro binders, but they show some problems. Organoclays are not suitable for feed ingredients due to toxicity of the interlayer quaternary alkylammonium ions, and carbons is a non-specific sequester, so this reduces possibilities for its practical application. Okay, another interesting product that can be used as a multi-toxin absorbent is activated clay. In the European project, big European project, MycoTi, we obtained this new product selecting raw bentonite and activated and functionalized this raw bentonite with an acid with an organic modifier in order to obtain a synthetic new materials that was then in vitro and in vivo tested. Concerning in vitro efficacy assessment, using a very low adsorbent dosage of this product, 1 kiloton, as you can see from the red bars, activated bentonite showed a higher mycotoxin adsorption values comparing with the native bentonite. These results were confirmed also by adsorption isotherms studies. Here you can see an example of an adsorption isotherm using the Langmuir mathematical model. And we found that for this activated clay, maximum adsorption capacity values determined for aflatoxin B1, fumonism B1 were higher than values found for zearalenone and ochratoxin A. Another interesting studies on this product was the desorption studies. Desorption studies consist in an adsorption at pH 3 and then in a desorption at pH 7. In this study, we performed also a desorption in a strong organic solvent to understand the strength of binding between activated clay and mycotoxins. As you can see from the red and green bars, a change of medium pH from acid to neutral did not release the adsorbed toxins. And in particular, aflatoxin B1 and fumonism B1 stay kept bound on the minerals also after an extraction with methanol. This to underline the good binding between this activated clay and mycotoxins. This product was also tested in vivo using two different animal models, rats models and piglets models, and using two different experimental designs. Rats were divided into two groups, one treated with activated clay and the other one untreated with activated clay. A single intragastric bolus administration of each mycotoxins was given to the rats, and urine samples at different time points were collected to perform a toxicokinetic studies. Concerning piglets, we gave a single intragastric bolus administration of a mycotoxin cocktail of aflatoxin B1, ochratoxin E, fumonism B1, and zearalenone, and dividing again piglets into groups, treated and untreated with the activated clay. In this case, blood samples at different time points were collected, and also in this case, toxicokinetic studies were performed. The analysis of mycotoxins were performed using the biomarker approach. Okay, what about results? Concerning these results, we studied what's happened at at least two important toxicokinetic parameters, such as AUC, area under the curve, and maximum urine or blood concentration of mycotoxins. Concerning rats, we found in the AUC a statistically significant reduction of the AUC curve comparing control and treated group for aflatoxin M1, ochratoxin A, and fumonism B1. We didn't find any AUC reduction for zearalenone and metabolites, but we found a statistically significant reduction in the Cmax values, another important toxicokinetic parameter. So we can see that this product, activated clay, is able to reduce bioavailability of mycotoxins in rats. Concerning piglets trials, was found a statistically significant reduction in aflatoxin B1 for aflatoxin B1 and ochratoxin A values in the AUC values, comparing control and treated group. And a trend reduction was found also for fumonism B1 and for zearalenone glucuronide, that in piglets' blood is the biomarker of zearalenone. For zearalenone, no AUC reduction was found, but was found a trend reduction for the Cmax values. How we can put together these trials, in vitro trials and in vivo trials of this activated clay? We can say, I would to say that first of all, before to try in vivo product, we need to have a very good mycotoxin binders in vitro. As you can see, this product was able to absorb more than 82% of mycotoxins, of this mycotoxin tested. And concerning in vivo, despite to the different experimental design between rats and piglets models, results were quite similar for these four mycotoxins. And I would say that its rats models can be a good model to test a prior product in vivo before to go to other more complex and with difficult management animal models such as piglets or other kind of animals. Okay, concluding my talk, I want to summarize these results, and you can see that majority of adsorbents except activated carbons are ineffective to adsorb deoxynivalenol. Each cell wall-based products adsorb preferably zearalenone. Agricultural byproducts such as grape pomace or durian pill modified are quite effective for aflatoxin B1, zearalenone, and ochratoxin A. Natural clay minerals adsorb preferably aflatoxin B1 at pH 3 and 7, and ochratoxin A and fumonidine B1 at pH 3. Organoclays can act as multitoxin adsorbent, but they are toxic, so this reduces possibility to for application in vivo. And activated clay can be a promising potential multitoxin adsorbent to be used in vitro and in vivo for different animal species. So I want to thank you for your kind attention. If you have some questions.
Guillermo, we cannot hear you.
You are on mute.
Oh, sorry, I was in mute, talking to myself here. So we have the question area on the side. You can open that question box. Just type it in, send to us. It can be in Spanish or Portuguese or English. We can translate and ask the questions. The answers, though, will be in English, but we can send on an email as a follow-up in Spanish and Portuguese the answers as well. So I will give a couple of minutes for questions as I start to receive them here. We will have another 15, 20 minutes for that. Okay. Let me ask the first question to Dr. Olga Verkieva. How to compare the different mycotoxin binders in the market?
It's a very relevant question. A lot of customers have issues to compare different products. And the most available tool is to compare in vitro efficacy, but I would not really recommend to use it as the only tool to compare products. The best, of course, if you can test your product in your animals. If you cannot do this, I would advise to request as much data, research data from the producer of the product as possible. And please focus on mycotoxins which are relevant for your situation. If you have deoxynivalenol issue, you can't use data on aflatoxin studies. Or if you have pigs, you cannot always rely on data that you get in poultry, especially in specific mycotoxins like zearalenone, which has different metabolic pathways in different species. In vitro binding data could mislead you, and you could see it on one of the latest slides of Dr. Vito, that there was a binder binding zearalenone at 99%, but when it was tested in vivo, it was not effective at all. So it's always important to have in vivo proof.
Okay, thank you. I have a question here to Dr. Julia. What about phytogenics to recover liver or other tissues damaged by mycotoxins?
Yes, that's very good questions. They are really important. But first of all, we need to eliminate the mycotoxins from the body as much as possible. And after then, we can start taking care of the liver and all other organs we can support during this mycotoxicosis. Because even in good cases, we cannot adsorb 100% of mycotoxins. For example, even when we speak about aflatoxin B1, we know that in vitro studies adsorption is more than 90%, even 100%, but we will never see the same in vivo, meaning that maximum 80% would be able bentonite would be able to adsorb, for example, aflatoxin B1, and the rest will go to the liver. And in that case, we will definitely need something to support liver, to help liver. So phytogenic in that case would be helping a lot.
Thank you.
We will have another session, one of the 5 we have for this set of webinars, which will be also devoted to the organ support. And I think this question will be even more answered during this session in April, I believe.
Should be in the end of April.
Next question, Dr. Olga. Is there a standardized EFSA Efsa test for mycotoxin binding, which is accepted in other jurisdictions?
This is available only for aflatoxin, and this is standardized for binding aflatoxin by 200 grams of bentonite per ton of feed equivalent at pH 5. And this is available in the European legislation for bentonite with number 1060 from 2013, I think. So this method is described in the law.
Okay. Thank you. Next question can be to either of you. Can you please talk about different methods of activation? I think it means about the clay activation.
Olga, would you start?
Yes, probably I will do. There are many different activation processes, and the most known is the process of activation of natural clays with cations. That, for instance, bentonites in nature, they are existing mainly as a calcium bentonite. But calcium bentonite is not very good in binding capacity for aflatoxins, for instance. And producers of bentonite, in order to approach the feed industry or mycotoxin binding binder producer companies, they are activating calcium bentonite with sodium salts. And sodium has more potential. It charges the layers of clays with more negative charge, and it's more potential for aflatoxin binding. Another way of activation, this is also extreme, I would say, it's activation with organic molecules like quaternary ammonium, or they also called quats. And in some markets of Latin America, these organoclays are also existing. They are the same principle like sodium cations, but they have much stronger positive charge, and they are bigger molecules. And they are resulting in very, very high adsorption. And they can adsorb a lot of molecules of mycotoxins, but they are non-selective. They can also do vitamins, minerals. But the problem with these organoclays is they are toxic because quaternary ammonium is a detergent. It's used for washing powder or some disinfectants, which are not allowed for use in feed. There are some patents available for activation of clays with, for instance, choline chloride, which is organic molecule, but it's more gentle. It's not toxic to animals, can be used in feed. And many other molecules could be potentially used for the activation, organic or inorganic molecules.
Okay, thank you. Next question. Will minerals in gastrointestinal tract interact with mycotoxin binders? Bentonite, activate charcoals, and others. Dr. Julia, want to answer that?
Yes, that's a very good question, and it's very often asked. Of course, any clays, any compounds will interact with nutrients, with vitamins and minerals, and even bind to some extent. But in the levels, the most of products used, like we used bentonites, the adsorption of the vitamins and minerals will be not significant, meaning that it will not interfere with the main function. And speaking about the activated carbon, it should be used in high levels, and it's like more binding capacity is more potent on this compound, and meaning that the binding capacity against vitamins and minerals will be higher. And of course, most of the experiments with the binding of vitamin and minerals is done in vitro. And you can find very nice reviews, even from the recent papers, showing that the clays can bind very good vitamins and minerals. But again, you need to understand that it's difficult to compare results in vitro with real results in vivo. Even with the same pH level and so on, the conditions will be completely different with different many other parameters with influence on these things. And for example, we have nice results showing that mycotoxin negatively affect antioxidant system. And we measured level of vitamins in blood of piglets, for example, and seeing that the level of vitamins was significantly decreased when the mycotoxins were in feed. And then when we used mycotoxin management product, level of vitamins was significantly higher, and even higher than in control, meaning that there is, if even there is some adsorption, but that the level of that adsorption is not significant, and it's not interfering with all the function with antioxidant system in this case. So, Olga, if you'd like to comment more.
The binding capacity or binding affinity towards different vitamins also depends a lot. And the most sensitive would be probably oil-soluble vitamins and some vitamins of B group, maybe B1, B2, but not all of them. But indeed, the in vivo proof showing that there is no interaction levels in blood, for instance, like Julia was mentioning, this is a very good confirmation that product is friendly to premixes, to essential nutrients. But of course, products which have already in vitro binding capacity very strong, and they can bind non-selectively mycotoxins, that there will be already a risk that they can bind also some vitamins and minerals. So it's a balance. Let's say you can't say that product does not bind anything, that probably it will be not effective against mycotoxins. And if it binds everything, that it will be probably binding also essential things.
Yeah, I agree. It's all about balance.
Okay, thank you. Next question here is inclusion of mycotoxin binders. Can you talk about inclusion of mycotoxin binders and if there's a limit of how much clay you can use on a diet?
I will take this question. It depends again on clay, and depends also on the legislation in the country. Like for instance, for a European community, for bentonite there is a certain limit, like not more than 20 kilo per ton of feed. But 20 kilo, it's a lot. So normally the binders are used at maximum 5 kilo per ton of feed. For other clays, as far as I know, there are no limitations, but I don't know by heart how FDA limits the use of clay. But I would say if you use binders or clays for whatever reason, maybe not for mycotoxin binding, if you use it below 5 kilos, I would say there will be no worry. And if you have a worry, it will be maybe around the nutrient binding.
Okay.
But in small levels, it would be no problem.
Yeah.
Even I recently came across the article from human medicine, and they are using cepiolite. By the way, cepiolite is also part of our products in most countries of the world. Cepiolite is used in human medicine as a carrier of some nutrients for slow release. So even it's beneficial, in fact.
Interesting.
Okay, thank you. Next question. What about T2? They act by contact. What's the most important mode of action of this product with T2? Julia, do you want to answer? Olga?
Yeah, I will start and Olga then comment. Yes, sure. For T2, especially in poultry, we can very often see these oral lesions, and meaning that it's so difficult to act in this case when it starts acting by contact already in oral for taking leading to these oral lesions. But again, you need to make everything in order to get the mycotoxin from the body and then support the digestive tract and support the epithelium as much as possible to decrease this negative effect. And we have even excellent results with T2 toxin when we saw this a very like severe case in the field in layers, and then we used our product, and in 2 weeks the oral lesions were significantly decreased, meaning that even if we cannot remove from the mouth from the beginning of T2, we can even with other means we can decrease this negative effect.
Yeah, it's important to know that binding is happening only when feed is mixed with gastric juices. So in the liquid surroundings. So in the mouth, there is already the first contact, and it's quite naive to believe that we can bind or any product can bind T2 already in the mouth, in the feed, which is mixed with saliva. But there are some other modes of actions. For instance, some products contain ingredients which can be also circulating in the blood, and then they increase the recovery of epithelium also in the mouth. And as Julia mentioned already, we have field evidence. We can't say that we can really protect 100% from mouth lesions, but we can reduce, notably reduce the appearance and severity of mouth lesions caused by T2 toxin.
Okay, thank you. Next question. The speaker mentioned about 30% to 40% adsorption of formalin B1 and zearalenone. Is that enough binding? And how can I evaluate commercial toxin binders efficacy in chicken and swine feed?
I will take this question probably. I don't remember exactly when it was the slide for in vivo or in vitro. I believe that was in vivo binding.
In vitro.
In vitro, yeah.
In vitro.
But even if it's in vivo or in vitro, doesn't matter. We can't expect 100% protection for any mycotoxin, even aflatoxin, which is the easiest to bind. In some animal species, we can bind up to 80% or give a protection up to 80%. In some species, it's not possible, like in swine, because it's very fast going to the bloodstream. Same for zearalenone. It's a molecule which is absorbed already in the smaller intestine and the upper parts. It will, some zearalenone will escape from the binding and will go to the blood. So, of course, when you have high levels of zearalenone, you can't believe that you have a product or you can find a product will protect your animal 100%. So what I would advise best to choose a product with the proven efficacy in vivo, because in vitro you also could see on the slide that 99% in vitro was resulting nothing, almost nothing in vivo, in pigs and rats. In rats was something like 12% reduction, in pigs was 0. if you found such product, you play with the dosage. The higher the dosage, the better would be the protection. If you find the effective product showing you at least 50% of reduction in bioavailability. And my personal advice nowadays that there are some bolus or toxicokinetic studies are available for testing products in vivo. So you give the bolus to the pig or chicken with mycotoxin and with the product. And you see the dynamic of mycotoxin and metabolites in blood. If the binder is able to bind mycotoxin, you will see the blood levels lower than in the control. So Vito was showing this type of trial. And that would be interesting for you for assessment of mycotoxin binding product. It will be more expensive than in vitro data, but it's much less expensive than the really performance trial through several weeks, because this trial takes a couple of days only.
Mhm.
Okay.
And may I add something? We had this, the bolus trial, as Olga mentioned, in piglets in Don, studying the pharmacokinetic of Don. So it was very interesting to see, and to see how fast is going to blood, and how fast it's neutralized by the product. So if possible, yes, it's this kind of trials can easily show how it's going.
There are several facilities are available in the world, and the methods are mainly based on the publication of Dr. Matthäus de Vries from Ghent University. He published his paper in 2012, and his method was applied in Ghent University, and you can test it there, or a producer of the additive can test it there. Also, INRA Toulouse, Isabella Swalt can do this, and Giuseppina Avantaggiato is a co-worker or partner of Dr. Vito, who was presenting today. They are also doing these models.
Yeah, in ISPA,
Perfect.
Italy.
Thank you very much. Okay, so we are about a minute to finish. We unfortunately have no more time for questions and answers, but everybody that sends the question, we'll try to do our best to send the answers back on an email and a follow-up. Margot is just sharing a link to fill up to fill up a form, just to let us know how everything went for you in terms of this webinar, if it's interested, and of course to receive information on the next series we will have. And I will give a couple of minutes as well to Mateus to say a few words to the Spanish speakers. So thank you very much, everybody here in North America and South America, and see you next time.
Ok, obrigado, Guilherme, obrigado, Olga e Júlia. Gostaria apenas de agradecer também aos clientes, convidados que estão presentes, e dizer que para a gente seria muito importante se vocês entrassem no link que a Margot compartilhou, para que possam passar para a gente suas impressões sobre o webinar. Então, obrigado a todos pela participação, espero vê-los novamente nas próximas sessões.
Obrigado, gracias, thank you. See you next time.
Thank you.
Goodbye.
Thank you. Bye-bye.
Bye-bye.